am thrilled today to be hosting Jonathan Lim, the CEO of Erasca. Jonathan, thank you for being here today.
Thanks, Jorge. It's awesome to be here.
Why don't we start at the top here, which is on you and your background. So you are the CEO of Erasca. You didn't start out on the business side. By background, you're a physician, and if memory serves, you were a surgeon. So walk me through the trip from the operating room to the boardroom.
Yeah, I grew up in San Diego just loving the movies and thinking maybe I'll be a filmmaker one day, but I also loved science and wanted to be a doctor and really use science to help people. So I was pre-med at Stanford, went back to Montreal where I was born, and then went to McGill, and then went straight into general surgery at New York Hospital. And I just loved it. So I was a really happy surgeon. But I did a 2-year break in Boston, and I worked at Dana-Farber Cancer Institute. So I did some cancer epidemiology research. Then I also did an MPH in healthcare management at Harvard School of Public Health. So that was my period of exploration. And during that time, I really decided, wow, entrepreneurship looks really cool. So I went to McKinsey for a couple of years.
Oh, man.
And then
down the dark path.
I entered the dark side. And then I joined Halozyme when they were a 4-person startup in 2003.
That's a couple of interesting jumps in what you just described. Being a practicing surgeon to a McKinsey, it seems like a leap, but in some ways those are pretty safe bets, right? From there, though, McKinsey to a 4-person startup. Talk to me about that leap, because that seems like the riskiest of the leaps from what you've described.
I'm a big believer in that things happen for a reason, based on my own Christian faith and other things. But my dad introduced me to one of his patients who had a venture capital startup, and her name's Audrey Viterbi. I met her, and she saw my background in surgery and McKinsey, and she thought, hey, you could be a CEO in biotech. And I thought she was joking, but then I got a call just a couple months later saying, hey, do you want to move to San Diego? I said, why? And she said, we have this biotech startup that we'd like you to run. And so I was
debating. Got called in as the CEO.
I got called in as a CEO. So I was 31 at the time. I knew nothing. I mean, I knew a little bit about biotech because at McKinsey, I served a broad range of clients across healthcare. But I mean, this was totally a leap of faith. And I was employee number 5. I didn't know what questions to ask. I didn't know what I didn't know. So we had 3 months of cash on hand, a company that had 0 VC dollars invested. And we said, well, how do we go public? And so the idea of a reverse merger of Halozyme, the private company, merging with a public shell company. So we were on the over-the-counter bulletin boards, and we traded for a few months. We uplisted to the American Stock Exchange about a year later, and then to the NASDAQ 2.5 years later, and then the rest is history. So yeah, really humble origins.
Wow.
Yeah.
And what was the stage of the company when you did this?
We were preclinical. So
you were a preclinical
we
company
really believed
in 2003 without
this was
VC.
on enhanced technology. I mean, the technology itself, we knew it would work, but it's just we needed people to believe in it. And so we were able to then really de-risk the technology, and we signed a deal with Roche. So they took a leap of faith where we got that deal done in 30 days at the end of 2006.
Wow.
And then we became the 4th performing stock in the Wall Street Journal that year, and it was just amazing. Like all of a sudden we were on people's radars, and we struck deals with Roche and Baxter and a whole number of other companies for enhanced technology, which now Halozyme is guiding to about $1 billion in revenue in the next few years.
That is a remarkable story. And then from Halozyme, what was your next leap?
Then I started my own fund. It was a family office fund called City Hill Ventures, and this was the idea of found, fund, and lead different life science companies. So sort of took a broad view of the healthcare industry, did some medical device, did some healthcare technology, did a lot of biotech, realized biotech really was my sweet spot. And so I started a number of companies, including Eclipse Therapeutics, which was a spin out from Biogen IDEC. Started Ignita later that same year. Joined them full-time in 2012 when Eclipse was acquired by Bionomics. So it was good early proof of concept. Wrote an article on it called Small Ball Investing. And then I ran Ignita for many years. I then started Bonti in 2015, which was a sort of a spin out of technology from Allergan. And then both Ignita and Bonti were acquired in 2018 in the same year. So that was a big year for City Hill.
So let's talk briefly about the Erasca story. We're obviously fortunate to have been early...
Thank you for being there in the early days.
Early stage investor in the company. It's been remarkable to see that company's journey. So Erasca, the company's tagline is erase cancer, which is really where the name Erasca comes from.
Right.
Let's talk generally about cancer. And I want to go back and talk more about the biotechnology industry and your journey as an entrepreneur. But if we just focus and zoom out for a second on the cancer landscape, where do you think we are in that journey?
So Erasca's mission, it's actually a portmanteau with a double meaning. So one is erase cancer, the other is to eradicate RAS-driven cancer. So that's sort of embedded in the acronym, so to speak. But I would say we're making progress. We definitely are not where we need to be, but we're still early innings. And I think what's really encouraging is where we are today is significantly better than where we were 5 years. And so with every 5-year increment, we just keep making progress. So it's just amazing to see the progress of immunotherapy and just how many therapies we have at our disposal for different solid tumor types and liquid tumor types for that matter. As you think about the monoclonal antibody approaches, the even T-cell therapies, CAR T, other aspects, there's a lot to be excited and grateful for. But I think what's most exciting is, especially with the checkpoint inhibitors, where you see the long tail. So you see about 20% of patients that have long-term remission that actually turns into cures. And I think the goal of the cancer fighting community has been, how do we increase that long tail? So rather than 20% or 1 in 5 patients, how can we get 2 in 5 or 3 in 5? And so we're exploring a number of combinations. But then with targeted therapies, there's some really exciting breakthroughs, whether it's some of the ADCs like in HER2, where you see just remarkable long-term survival outcomes, OS and PFS with in breast cancer, and now they're in 5 different indications, including tissue agnostic. And I think we're scratching the surface of the combinations that we can explore.
I put this out as a sort of a straw man argument. One would think that if cancer is primarily a disease of DNA, as we've gotten better at being able to sequence tumors, to sequence cancer, to analyze it, how much has that illuminated our ability to figure out how to target cancer, how to find its Achilles heel? In other words, if we can understand cancer drivers, and if most cancer drivers are genetic in nature, can we work backwards by analyzing the genetics of all cancer and then come up with strategies for targeting tumors in a way that will actually help us beat cancers? Just
a few years ago, I learned about ecDNA. So extrachromosomal DNA are these circles that basically tumors use to harbor massive oncogene amplification. And so most of the industry didn't even know that this circular chromosomal DNA was a mechanism for driving cancer as well as metastases and recurrence, until Paul Michel and others identified this and developed diagnostics to look for it. And so that's some of the work that we're doing at Boundless Bio, which I co-founded with the folks over at Arch and with Paul Michel. And we're really excited about that approach of looking at the genetic vulnerabilities of these ecDNAs and then using that to combine with some of the more traditional approaches as well.
Yeah, I think that's what's interesting and probably also frustrating about our field, which is the biology, as proves to be the case again and again, is always more complex than we originally think.
Right.
So we think, okay, we can sequence tumors, we'll figure out where the mutations are, we'll figure out how to target them. But then we learn about things like ecDNA that just obviously complicates the picture.
Right.
On the one hand, in terms of figuring out what to go after. And on the other, one of the things that I think is reason to be optimistic, but there's still a long way to go, as you say, we're still scratching the surface, is we get increasingly better and more sophisticated in the types of tools or the modalities that we develop to hit cancer with. The weapons we have against cancer, that armamentarium, that weapons chest is expanding, continues to expand. And so I think there's reason to give hope. If we were to flash forward 5 years from now, what would be your prediction on what will have proven to be the next sort of set of great leaps forward?
Well, I think we're learning more and more. I think we're taking new chemical modalities and approaches to shutting down different oncogene drivers or quote unquote undruggable targets. So I think the modalities are really exciting. Even in our own experience at Erasca, Revolution Medicine here in town is a pioneer in the approach of taking a molecular glue or cyclophilin A binder to drug RAS, which RAS in the active GTP state has historically been really difficult to shut down. But we were really just so encouraged by their data. This was about a little more than a year ago, regarding how they could take RMC 6236 and shut down RAS in the active state and show responses in G12D and other types of tumors. And so we actually have our own cyclophilin A molecular glue called ERAS-15 that we just in-licensed, and we're really excited about that with even higher potency than RMC 6236. But it's approaches like that I think are really moving the needle, whether alone or in combination with other approaches. And it's what we need is to keep advancing the standard of care. Until RevMed showed that you could target RAS wild type, I actually was very skeptical that we'd be able to do that. But then when I saw the data, I was just amazed that this was safe and reasonably well tolerated. So it's really exciting to see the amount of progress that's happening with new modalities.
And I think the kinds of things you described, like these sort of leaps forward, give us lots of reasons to be optimistic.
Right.
That the next 5 years and beyond are going to continue to evolve in a way that will help us make great improvements in this field and in other disease areas as well. Shifting gears a bit, what'd you do this summer?
Well, one of the highlights is my family went on a trip to the Galapagos Islands. So this was basically a Stanford travel study program with Bill Durham, who was a faculty member. He taught in the human biology department at Stanford for decades. And this was on our bucket list to go on this trip. And it was just a once-in-a-lifetime opportunity.
And Galapagos, of course, are the islands where Charles Darwin
That's right.
Sails down there and has the sort of eureka realization around
natural selection, and his whole theory is born there. And believe it or not, I mean, he was in his 20s. I think he was 26 or something when he discovered this. And he was only there for less than a month.
So Charles Darwin goes down to Galapagos, spends some time there, and comes up with the theory of evolution, with the concept of survival of the fittest. You go down there this summer. You spend some time in Galapagos. What theories did you come up with?
There's this bird called the frigate. And so most birds that dive for fish, they have oil glands that basically, cover their feathers and allow them to not sink. Mm-hmm. Now, the frigate bird has vestigial oil glands, so they can't do this. If they fish on their own and dive bomb, they're dead. And so
because they'll sink?
They'll sink, yeah. And so they've basically evolved to be bigger than all the other birds. So when the blue-footed boobies and the pelicans and the other birds dive bomb and get the fish, the frigates come in and they just take the fish away from them. And so they're called kleptoparasites. But there's this whole ecosystem of these bully birds.
And do they fight them in the air, or do they fight them on land?
They fight them in the air. So they basically just intercept and take, they basically wrest the fish away from the other birds. And so that's just evolution at work. And then, in terms of the flora, there are these trees that are called scalesias. They're named after one of the scientists. And if you look closely, the trees they're beautiful. They almost look like Dr. Seuss types of trees because they're just so exotic. They're actually daisies if you do the genetic analysis. And so how do daisies become trees? Well, it's, again, very Darwinian in terms of, herbaceous plants sort of fighting as bushes for sunlight and scarce resources. But these daisies just keep growing, and they grow as far as they can and become trees just so that they can outcompete the other the other plants. And so when you draw the parallels with our industry, I mean, clearly, some of the lessons are you adapt or die. So if you don't learn to adapt in our industry, you're not around very, very long. And the last few years, I think, have been sobering for all of us, Erasca included. It's just been really tough and very Darwinian in terms of competition for attention from investors, attention from partners, attention from employees, in terms of the war for talent. So it's just been really tough. And so you have to adapt or die. And so we've done a lot of pivoting in terms of prioritizations and reprioritizations and trying to get access to capital and trying to have programs that people really care about and that can really move the needle for patients. And then the rest takes care of itself. And I think everyone's trying to do that. But then it's really the realization that we all need to embrace change and embrace it, not just to survive, but to thrive. So in addition to defense, there's also offense. So at Erasca, we're focused on the RAS-MAP kinase pathway. Our lead program in the clinic is a compound called naporafenib, which is a pan-RAF inhibitor. And we're super excited about that for NRAS mutant melanoma. But NRAS mutant melanoma, there's about 50,000 patients total in the US. It's definitely an unmet need, but it's not a massive one. And so we were looking at different programs that could really move the needle for hundreds of thousands of people in the US and globally. And so we were able to in-license successfully two RAS targeting programs. One is a pan-RAS molecular glue, as I mentioned earlier, and then the other is a KRAS selective pan-KRAS small molecule inhibitor. And so we're really excited about those programs and the ability to combine with standard of care in pancreatic cancer, in lung cancer, colon cancer, other solid tumor types, and then with each other. And then if you combine it with the molecular glue for any acquired resistance mutations, then that could really be the nice interesting combo.
If I'm a founder, if I'm an entrepreneur today, and I'm sort of navigating through what is a very difficult environment, a very inhospitable environment, what are some of the adaptations that I should have front of mind?
Yeah, no, I mean, I think I would say that don't be afraid to pivot. And if you're going to pivot fast. So the faster you make the decision, the better. All the companies I've been at, there's always been one or multiple pivots. And so at Erasca, we actually have had sort of three epics at the company. So in the early days, we were focused on discovery for new RAS targeting agents. And then we sort of brought in a number of assets and we went public, as you'll recall, around the ERK targeting program. And then we brought in naporafenib from Novartis, and that's a really exciting program. But I think high probability of success, but in terms of moving the needle on a massive unmet need, probably not as much. And so then we brought in the latest, which are these two RAS targeting agents. And now the enthusiasm amongst the buy side and sell side is really very high. So I think we're in the sweet spot right now. And so it's really sort of embracing the uncertainty, but applying creativity, resilience, and decisive action to navigate that.
Yeah, and I think that's a key point, right? Because pivoting is daunting. So now, if we look at the landscape broadly, it's a challenging environment for all the reasons we've said. Is there a silver lining? Is there anything out there on the landscape that you'd say, look, this is actually, these are calm seas to maybe head towards if you're an early stage company? The answer may be no.
Yeah. No, I mean, I think the pendulum always swings back and forth between platforms and products and back again. I think we're back in the product cycle where I think really focusing on what is that product or therapeutic that's really going to move the needle for patients. And so there's a number of AI companies that are really building that drug development expertise to take a product or products further into the pipeline to be able to generate the clinical data that investors and partners want to see. So I think that's really the playbook at this point. In this environment, people want to see what impact can you have on patients, whether it's discharging safety or showing efficacy proof of concept. I think it's really important to show what your therapeutic can do. And then it's really just marshaling the resources to focus on that, on what's really going to move the needle. And by the way, when the pendulum does go back, raise money, but really treat it as if, you're in a bear market as opposed to a bull market. So I think, one of the things that we did right at Erasca was when we went public in 2021, day one after the IPO, we said, now that we have all this money, what are we going to do with it? We're not going to spend it. We're actually going to save it. And so we knew that the pendulum was going to swing back. It was going to be tougher days ahead.
So we've talked about pivots. We've talked about creativity. We've talked about pendulum swinging. One of the most remarkable pendulum swings, pivots that encompasses creativity, I think would apply to you. You said at the very beginning you loved Hollywood. You started your fund, City Hill Ventures, and you said it primarily invested in biotechnology companies. That is evolving.
Right.
So now you're investing in things that are very much not biotechnology companies.
Very Darwinian, isn't it?
It very much is. So talk to me a little bit about that.
Yeah. So, I was born in Montreal, grew up in Southern California. But in 2018, when Ignita was bought by Roche, it allowed me to take 3 months off. So I was off the grid. I put my iPhone in the drawer. My wife got me a flip phone with a different number.
You had a burner?
I had a burner phone.
Sweet.
Yeah, believe it or not. I still have it, but I don't use it anymore. I just prayed. I reflected. I spent a lot of quality time with family, friends, traveled, did a lot of creative pursuits. And so I came back in October. We hit the ground running with Erasca. And then later that year, I started a company called City Hill Arts, which is an offshoot of City Hill. And so the idea is to revitalize people, planet, and perspective. It was just this idea that can we shape people's hearts and minds in a powerful way with inspiring stories, hopeful stories, stories of love, kindness, stories that bring people together and unite us and show us what unites us as opposed to divides us. And so out of that came this slate of movies that embody those kinds of stories. And so we are genre agnostic, but really the unifying theme is what unites us and stories with heart.
So you make movies now?
I make movies now, yeah. So
I can't tell you how cool that is, by the way.
Thank you. No, I feel so blessed to be doing what I love in terms of the twin passions of science and biotech and arts in the movies.
So I'm going to attempt to use Hollywood industry terms, but what's on the slate of movies? What's on the slate? Is that even how you pronounce it?
Yeah, that's it.
Okay, so what's on the slate in terms of movies that are coming down the pike?
So the first big movie for us is showing in theaters now. It's called My Penguin Friend. So it's a story inspired by a true story between a Brazilian fisherman that came across this Argentinian penguin all covered in oil. The fisherman nursed him back to health, and then every year the penguin swam thousands of miles from Patagonia to this fisherman's remote village in Ilha Grande, Brazil. So that happened every year for 8 years. That's the true story. It blew up on social media in 2016. And then one of our producers tracked down the real fisherman, and he basically got his life rights. And we cast 10 real penguins in the role of Dindim. So about 80% of the movie is shot with real penguins. You can't put penguins in peril, though. So 15% is with CGI, and then 5% is with puppets or animatronics.
Oh, wow.
Yeah.
Well, first of all, you talk about stories that unite. You're telling the story of a Brazilian and an Argentinian getting along.
Hey.
So that's amazing in and of itself.
That's a miracle, right? But that's the first. And then we have another movie, a small movie called The Secret Art of Human Flight. So it's about a widower who is grieving, and he meets this guru who teaches him how to fly.
Oh, wow. And is that also a true story?
No, it's a grounded sci-fi, but it's going to be a day and dates release actually this Friday. So people could see it in the theater as well as on streaming at the same time.
Oh, my goodness.
Yeah.
You guys are busy.
So, yeah. And then, we've got some other movies that we're working on.
Is there a comparison between the biotechnology industry and movie making? Because at the surface, you would think these feel pretty similar. You're making an investment upfront in the hope that you have a hit.
Right.
Yeah. a hit is used both in biotech and in movie making.
There's actually an uncanny parallel between those two industries. So let's say a given project in the movie business, let's take a really successful film, The King's Speech, won the Oscar. How many years do you think that took to make?
I'm going to guess a long time. I'm going to say 10 years.
Yeah, 13 years.
My goodness.
Yeah. So they call it, in Hollywood, they call it development hell, just because it takes so long to get these movies made. So in our industry, it's about 8 to 10 years for a typical drug to make it from the early beginnings to launch. And in Hollywood, it's no different. I mean, there's a lot of nonlinear paths that these movie projects take, and there's a lot of stakeholders. And then the probability of success of any given project. So when you look at a drug, the POS really increases by stage. It's kind of the same in Hollywood, except that the outcome in terms of box office revenue and performance is really just imprecise. So there's no way to know whether a given project, I mean, I love penguins. You probably love, I don't know anyone that doesn't love penguins, but I still have no idea how well my penguin friend's going to do. And so you have to take a portfolio approach in the film investment arena as you do in biotech.
So we depend on hits. We each have our own version of development hell, right? In our case, it's discovery and development hell. Is Dim Dim still with us?
Dim Dim is not with us, but the 10 rescue penguins are. And in fact, 2 of them fell in love on set. And so, they were unusable because they would just stand back to back and stare off hopelessly in the distance.
That's fantastic.
So we actually had 8 penguins to work with.
Well, Jonathan, I want to thank you so much for spending time with us today, and really want to thank you more importantly for all that you do, not only for the magic of making movies, but all you've done for the magic of making medicines.
Oh, thank you very much, Jorge.